Hello, everyone. Today, we will discuss T-cell prolymphocytic leukemia. T-cell prolymphocytic leukemia (T-PLL) is a rare, aggressive mature T-cell leukemia characterized by systemic proliferation of highly malignant prolymphocytes. Despite its low incidence, which is less than 2% of all mature lymphoid leukemias, T-PLL presents disproportionately high clinical challenges due to poor responsiveness to conventional chemotherapy and limited availability of disease-modifying treatments. In this report, we consolidate current insights into T-PLL's clinical complexity and developmental landscape. While therapies like Alemtuzumab offer initial response, relapse is common and durable remission is rare. A modest but expanding therapeutic pipeline now targets previously inaccessible molecular mechanisms such as MDM2 inhibition, HDAC modulation, and T-cell receptor signaling. Our integrated view connects clinical potential with unmet need, analyzing assets by clinical stage and strategic viability. T-PLL predominantly affects patients aged 60 years and above with a slight male bias. The disease is characterized by splenomegaly, lymphocytosis, hepatomegaly, and immune suppression. Median overall survival ranges between 12 and 24 months. TCL1A overexpression, ATM deletions, and aberrant JAK STAT signaling are among its hallmarks. While Alemtuzumab, a monoclonal antibody against CD52, remains the current frontline treatment, it offers high initial response rates, approximately 80%, but limited median progression-free survival of approximately 12 months. For most patients, T-PLL eventually relapses and becomes refractory to Alemtuzumab, highlighting an urgent need for alternative therapeutic strategies. Current treatment options face several limitations. First, lack of long-term remission strategies. Second, poor tolerability of current regimens, including Alemtuzumab and fludarabine-based chemoimmunotherapy. Third, limited options for relapsed or refractory patients. And fourth, absence of biomarker-driven treatment personalization. The landscape has begun to shift from a purely symptom management approach to one that embraces more precise and targeted interventions. Although no targeted therapies have yet reached the market, promising candidates are advancing through clinical trials, offering hope for more effective and tolerable treatments. In terms of pipeline landscape, as of 2025, over 15 investigational products are advancing through preclinical and clinical stages. Emerging approaches reflect a shift toward precision, oncology, and immunomodulatory strategies. Key target classes include the MDM2 axis, HDAC inhibition, BCL2 inhibition, JAK, STAT, and PI3K, AKT pathway modulation. Notably, advanced bioinformatics tools and multi-omics approaches are increasingly used to stratify patients and guide early-phase enrollment. Predictive markers such as p53 mutation status and TCL1A overexpression are under investigation to enhance therapeutic alignment. Moving on, let's look at clinical development by stage. Currently, T-PLL-specific phase 3 trials remain scarce. However, several agents are being evaluated in basket trials for mature T-cell neoplasms. These include biologics and targeted small molecules with endpoints centered on overall survival, objective response rate, and treatment-emergent adverse events. In phase 2 spotlight, APG-115 being developed by Ascentage Pharma is a potent, selective MDM2 inhibitor that has shown promising results in hematological malignancies. It works by blocking the interaction between MDM2 and p53, restoring p53 pathway activity. In our hematological malignancies, APG-115 has shown good cytoreduction and manageable toxicity. In a trial of 132 patients with T-PLL, the agent demonstrated an ORR of about 40% with an interim analysis. Disease control rate was more than 60% with main toxicities of thrombocytopenia and fatigue. A companion biomarker exploration is ongoing. In phase 1 spotlight, Romidepsin being developed by Celgene Corporation is a histone deacetylase inhibitor with immunomodulatory and cytotoxic activity. Previously approved for PTCL, its repositioning in T-PLL is under early exploration. The agent showed dose-limiting toxicities of neutropenia and QTc prolongation in early trials. However, it has the potential for synergy in combination trials. In preclinical and discovery assets, several agents target immune evasion, chromatin accessibility, and microenvironmental support of leukemic cells. These include PI3K-gamma inhibitors, Notch inhibitors, and engineered T-cell therapies specific to TRBC1. Next, we look at inactive or discontinued programs. A small number of candidates were halted due to unacceptable toxicity profiles, insufficient efficacy in early-phase cohorts, and strategic reprioritization by developers. Following this, we will discuss leading developers and competitive landscape. Several entities are involved in developing potential therapies for T-PLL. These include Ascentage Pharma, leading the development of APG-115 with a focus on biomarkers. Celgene, now BMS, repositioning epigenetic modulators like Romidepsin, and AbbVie, exploring BCL-2 inhibitors in T-cell neoplasms. Additionally, academic alliances are driving biomarker-driven and CAR-T studies. Partnerships and orphan drug designations are key growth accelerators for many of these programs. Finally, let's look at market forces. Several factors drive the T-PLL market. These include improved genomic profiling enabling targeted approaches, expansion of rare cancer drug incentives, and growing precision medicine frameworks in oncology. However, major challenges remain: limited patient pool and trial recruitment difficulties, absence of validated surrogate endpoints, clinical and biological heterogeneity within T-PLL populations, and high costs and long timelines for biologic development. In conclusion, the future outlook for T-PLL treatment is cautiously optimistic. Integration of real-world evidence, use of biomarker-guided design, and application of combination regimens represent actionable paths forward. Ongoing trials are anticipated to shift treatment paradigms within 5 years, especially with the entry of immuno-oncology agents and genetically engineered T-cell therapies. Strategic focus areas should include broader application of genomics in treatment selection, exploration of dual pathway inhibition, and improved access and equity in clinical trial. We hope you found this report informative. Thank you for watching.